A very warm good afternoon. I'll be speaking on this groundbreaking technology, CRISPR editing, and its applications in ophthalmology. And I would like to thank Dr. Rohit Shetty for giving me this opportunity to represent Nara and Netrala's work at this platform. So why are scientists so obsessed about editing genes? Because genes have a fundamental role in building and sustaining life. It is the dream of every scientist to be able to edit DNA because it will be like hacking the code of life and it has the potential to change our futures. We already have certain gene editing tools like zinc finger and talons, but they require a complete redesign of the protein DNA interface to target a gene. CRISPR, on the other hand, is very simple, effective, and it can cut with extreme efficacy. And unlike others, it is customizable. So think of it like editing an essay on your computer. You tell your word processor you want to replace this word or change this word, and the word processor will do it. It is as simple as that. So CRISPR is an acronym for clustered, regularly interspaced, short palindromic repeats. So it is basically a locus where there are DNA repeats and spacer DNA in between. So it has two key molecules. One is Cas9, which is the molecular scissor, and then we have guide RNA, which will tell the molecular scissor where exactly to cut. And it will monitor that it has been cut at the right place. So where was this originally found? Originally, CRISPR was a part of bacterial defense system. Bacteriophages are the viruses that infect the bacterial cells. So CRISPR is like immune memory. It prevents the second attack. So what happens is, when a virus attacks a bacteria for the first time, a part of its genome is chopped off and stored in spacer DNAs. So spacer DNAs, what you see here in multicolor, are nothing but the pieces of viral genomes that have previously infected the bacteria. Now, this coding strand of the CRISPR and the DNA repeats of viruses are now transcribed to mRNA, and they are cut into short pieces of RNA called the CRISPR RNA. These small pieces then incorporate into Cas9 protein. This is now good to go for the second attack. So whenever there is second attack, the guide RNA will tell the Cas molecule, okay, this is a foreign material, it's dangerous, and we need to cut it. This complex attaches to DNA the next time, and a double-stranded DNA break is introduced. How can we use it to our benefit? So we can, here at the cut part, we can insert another DNA. So what all can we do with CRISPR? We can actually target and replace genes in humans. It is already used widely in agriculture and food industry, and it has large potential in many types of cancers, diabetes, hepatitis B, hypercholesteremia, and another possibility is to edit germline cells. However, it may not be applied due to ethical issues. In ophthalmology, work is already being done in the field of retinitis pigmentosa, Leber's congenital amaurosis, proliferative vitreoretinopathy, and some corneal dystrophies. What we already have in the market is LuxAeterna. It is the only gene therapy approved by US FDA ever for any disease, any genetic disease, and it is for individuals with an inherited retinal disorder where they have mutation in both the copies of RP65 gene. But it's very, very expensive. One vial is 6 crores, sir? 6 crore for one vial, for one subretinal injection. It's that expensive. So many of us must be wondering whether it is safe. Yes, this is a recent trial published on 7th of February this month, which ensures safety in cancer patients without any serious side effects. So here, a lot of novel work is being done in Nara and Netrala's Grow Lab on CRISPR, and probably it's the only lab in India working on CRISPR in ophthalmology, and it has received a grant from Department of Science and Technology. And these are some of the pictures of the lab from Nara and Netrale, conceptualizing and designing and cloning the single guide RNA. They're also working on lentiviral and other delivery systems. As a proof of concept, Nara and Netrale is also trying to validate the results at a DNA and protein level. So the research question we had in the mind was, how and when can we use this CRISPR technology? So it can be used to repair mutational hotspots in diseases like Fuchs and keratoconus, and donor templates can be used for a desired change by knock-in. In the lab, we are also trying to target right sites such as mutational hotspots and essential exons in the genes for precise outcome. So how can we be sure that the gene is edited? So assays like T7 endonuclease assay help to identify the precise gene location. Now this is interesting part of the study where the after effects of gene editing like genotyping, phenotyping and the validation at protein level is also being done in the lab. So the graph here is indicating the reduction in the protein level after gene knockout. We are also improvising on vector delivery system to find more efficient ways to deliver this gene editing tools to the target cell. This work is already being done in Nara and Nethralaya. So here is a simple model. I'll explain you how a gene functions and how a disease model is created. So if you can identify a particular gene in keratoconus, the relevant model to test is a mouse or a pig, or if you want to check ex vivo, it can be induced into a pluripotent stem cells. So once a gene is identified, embryonic stem cells with targeted gene disruption, that is knockout using CRISPR, is injected into the blastocyst of a pregnant mouse. And by inducing environmental factors like eye rubbing or any other trigger like inflammation, you can have the baby mouse to develop keratoconus, and this will be the proof of concept that this particular gene caused keratoconus. So why is CRISPR so revolutionary? It is because it can be used to target any gene. It is customizable. Only change required is the change of the guide RNA. And it is possible to store all the CRISPR RNAs in a library, in a genomic library. So what is the future? Yes, it is the game changer in enhanced understanding of human diseases, and it can definitely revolutionize the human gene therapy. Thank you so much. And I thank the team led by Dr. Rohit Shetty and Dr. Arka for this amazing venture.
Thank
Any
you, Dr.
questions?
Shruti. Do we have any questions?
So, Dr. Shruti, thank you. That was excellently summarized. We are working at a CSIR lab. It's called the Institute of Genomics and Integrative Biology. And just wanted to add on to some of the experience we have with the CRISPR genomic editing. So while initially using HDR and NHEJ, we were seeing a lot of insertion deletions and off-target effects. Tried significantly on the base editing aspect, and that's where we realized that a lot of times you're not finding PAM, which is very close to the single nucleotide changes. Especially the editing window was very distilled to the PAM as well.
Yeah.
So very initial trials at prime editing now. November it was just described, and we've just used some of the reverse transcriptase along with the prime editors. And another interesting thing that we noted was the distribution of mutations. So we're largely working on RP, and the circle gene with especially a lot of mutations, for example in rhodopsin or so, that are described in Western literature is not what we are seeing over here. So that makes it very
difficult for
precision medicine based and very relevant to do this kind of work in India.
So if you
for our own
notice any
patients.
new mutations?
Circle is something, for example, I'm just is one of the ceramide kinase like gene. That's one of been the commoner mutations, the genes which is having mutations that we've seen, and especially a 2 base pair deletion and so. So prime editing in that sense has
Have you seen multiple mutations also in the same patient?
That's been uncommon, but yeah, have seen that as well. In a single gene, multiple mutations, be it both at the intron or the splice site mutations.
Thank you so much.